beginning of life, narrate the dawn of life on Earth with scientific accuracy and emotional storytelling. Highlight the formation of early conditions, the first molecules of life, and the miraculous emergence of biological processes. Hello everyone. Today we embark on a journey back in time to a world unlike anything we know, a world of fire, water, and swirling gases, where life, as we understand it, was yet to be born. Imagine a planet, young and restless, its surface molten, oceans forming in chaotic patterns, and the atmosphere thick with gases. This was Earth nearly 4 billion years ago, a place of extremes and possibilities, where the stage for life was being quietly set. Life did not appear in a single instant. It emerged through a symphony of chemical reactions. The early oceans, rich in minerals and energized by lightning and volcanic activity, became the cradles of creation. Within these primordial waters, simple molecules like amino acids, the building blocks of life, began to form spontaneously. These molecules combined in complex ways, giving rise to chains and structures capable of catalyzing further reactions, delicate dance that would eventually lead to the first self-replicating entities. As the planet cooled and chemical gradients formed along the shores and shallow pools, the conditions became just right for life's first whispers. Tiny, invisible molecules engaged in intricate networks of reactions, experimenting in molecular evolution long before cells, DNA, or even membranes existed. These were the precursors to the living world, a silent and invisible revolution that would ultimately reshape the planet. Scientists today look at these processes through the lens of modern chemistry and biology, imagining how hydrothermal vents, tidal pools, and mineral-rich surfaces could have facilitated the formation of protocells, the earliest structures that displayed some characteristics of life. Encased in lipid membranes, these protocells were fragile and fleeting, yet within them, chemistry began to mimic biology. Some molecules learned to copy themselves, others stored energy, and a basic metabolism emerged. Slowly, step by step, the first glimmers of life emerged from the chaos. This epoch was a time of experimentation. Every molecule, every reaction, was a potential path toward the first living entity. Amidst the volcanic storms and the pounding waves, something extraordinary was unfolding, the initial spark of life. Unlike anything mechanical or predetermined, life arose as a product of countless improbable events converging in perfect synchrony. Consider for a moment the enormity of this achievement. From lifeless rocks and water, a process so intricate and fragile emerged, giving rise to systems capable of growth, replication, and adaptation. It is here, in this chaotic crucible of heat, light, and liquid, that the story of all living beings, including Easterly, begins. The first life was microscopic, invisible to the naked eye, yet it carried within it the potential for an unimaginable future. These primordial entities were simple, yet remarkably resilient, harnessing chemical gradients, absorbing nutrients, and eventually evolving into more complex forms. Over millions of years, these humble beginnings would seed the oceans with life, creating the foundations for the first cells, the first microbes, and eventually the rich tapestry of plants, animals, and humans. We are, in a very real sense, the inheritors of that first spark, the outcome of a process that started in a world of fire and water. Driven by chemistry and sheer improbability, life's dawn was not marked by a dramatic explosion, but by a quiet, persistent accumulation of order amid chaos. And it is this persistence, this tenacity of molecules striving toward life that laid the foundation for every organism that has ever walked, swum, or flown across our planet. This is the story of how chemistry transformed into biology, how mere molecules organized into the first living entities capable of growth, replication, and adaptation. Imagine a tiny droplet, no larger than a speck of dust, floating in the primordial soup. Within it, countless molecules are busy interacting, forming chains, looping into structures, and experimenting with the laws of physics and chemistry. Lipid molecules arrange themselves into membranes, creating a boundary between the internal world of this droplet and the vast, untamed ocean surrounding it. Inside, nucleotides and amino acids interact, forming the earliest versions of genetic and enzymatic systems. This is the protocell, the precursor to all life. The first cell was incredibly simple compared to the complexity of life we see today. Yet it possessed all the essential features: a protective membrane, a way to store and transmit information, and basic metabolic processes to convert energy from the environment. Scientists believe that RNA, a versatile molecule capable of storing genetic information and catalyzing reactions, played a crucial role in this first cell. This RNA world hypothesis suggests that life may have begun as a self-replicating RNA molecule, later enclosed within lipid membranes to form the first true cells. Within these primitive membranes, a delicate balance of chemistry created stability amid chaos. Proteins emerged as catalysts, speeding up chemical reactions. Metabolic pathways formed, allowing the protocell to extract energy from the surrounding water, minerals, and sunlight. Every reaction was a step toward greater organization, a step toward life as we recognize it. The first cell was not a singular miracle, but a gradual achievement, product of countless molecular experiments converging successfully. Fossil evidence and modern microbiology give us glimpses of these ancient forms. Stromatolites, layered structures created by microbial communities, date back more than 3.5 billion years and suggest that simple cellular life dominated the early oceans. These first cells were resilient, surviving extreme temperatures, volcanic activity, and a thin, turbulent atmosphere. Over time, they evolved, diversified, and spread, setting the stage for more complex life forms. Think about the significance. A microscopic entity capable of reproducing itself, carrying genetic instructions, and interacting with its environment had appeared. This tiny cell carried the potential for all future life, plants, animals, humans. From this one moment, evolution began its grand experiment, slowly, over billions of years, shaping the incredible diversity we see today. The first cell symbolizes the bridge between chemistry and biology, the moment when the universe began experimenting with life in earnest. It was small, invisible, and fragile, yet it contained the blueprint for everything that followed. Without this fundamental step, there would be no oceans teeming with microorganisms, no plants carpeting the land, no creatures roaming the earth, and no humanity to marvel at the journey of life itself. This story of the first cell is a story of perseverance, of improbable success in the face of chaos, of molecules learning to cooperate and organize. It reminds us that life is not a random accident, but a process, a series of events that led from simplicity to complexity, from chance reactions to self-sustaining organisms. Every living being today is a descendant of that first tiny cell, a testament to the power of life to emerge, adapt, and endure. Today, we dive into the vast, ancient oceans of our young planetary realm that predates land, atmosphere, and human imagination. Imagine a world where endless stretches of water glisten under a dim sun, waves churn across volcanic shores, and deep, mineral-rich pools teem with the potential for life. This is where life truly began, and it is here that the first chapters of our biological story unfolded. Water was not merely a habitat. It was a chemical laboratory, a crucible in which molecules could collide, interact, and evolve. The oceans of early Earth were warm, shallow in some regions, and enriched with minerals from volcanic vents. They contained hydrogen, nitrogen, carbon compounds, and other elements essential for life. In these waters, the first organic molecules, amino acids, nucleotides, lipids floated freely, forming complex assemblies, testing combinations, and laying the groundwork for the first cellular life. The first life forms in these oceans were microscopic, invisible to the naked eye, yet immensely significant. Single-celled organisms, likely resembling modern bacteria, thrived in the nutrient-rich waters. These microbes were masters of their environment, capable of extracting energy from chemical gradients, sunlight, or even volcanic activity. Through photosynthesis and cheosynthesis, they began to alter the ocean's chemistry, gradually producing oxygen and changing the atmosphere, a transformation that would eventually make Earth habitable for more complex organisms. Fossil evidence, including stromatolites and microbial mats, reveals the incredible diversity and activity of early aquatic life. These structures, formed by layers of microorganisms trapping sediment, provide a tangible glimpse into a world dominated by single-celled life. They tell a story of survival, adaptation, and innovation, a story written over billions of years beneath the waves. Life in water was not uniform. It evolved in myriad niches. Near hydrothermal vents, extremophiles thrived in complete darkness, feeding on chemical energy released by the Earth's crust. In sunlit shallows, photosynthetic microbes captured sunlight, producing energy and oxygen as byproducts. This interplay between different types of early life forms created a dynamic ecosystem where cooperation and competition drove evolutionary progress. These early aquatic organisms were also responsible for one of the planet's most profound transformations, the oxygenation of the oceans, and, later, the atmosphere, known as the Great Oxidation Event. This process began with microscopic photosynthesizers converting sunlight and carbon dioxide into energy and oxygen. Over millions of years, these tiny life forms reshaped the planet, paving the way for the first multicellular organisms. Without the oceans and these pioneering microorganisms, life on land would never have been possible. The ocean was also a protective cradle. Here, early cells were shielded from the harsh radiation of a young sun and the extreme conditions of the nascent atmosphere. Water regulated temperature, moderated chemical reactions, and provided a medium in which complex molecules could move and interact freely. In essence, the oceans were a womb for life, offering stability, nutrients, and the possibility of growth. As life thrived underwater, complexity slowly increased. Cells began to aggregate, forming colonies. Specialization emerged. Some cells took on roles in energy production, while others focused on reproduction or defense. These early communities laid the foundation for multicellularity, a concept that would forever change the trajectory of life on Earth. Consider the incredible resilience and adaptability of these early aquatic organisms. They survived volcanic eruptions, fluctuating salinity, temperature swings, and the violent churnings of tidal pools. Each survival, each adaptation, was a step toward more complex ecosystems. Predation, symbiosis, and competition concepts we often associate with later animal life were already at play, albeit in microscopic form. By observing these ancient oceans, we gain insight into the profound creativity of life. What began as simple chemical reactions became self-sustaining, adaptive systems capable of responding to environmental challenges. The story of life in water is a story of perseverance, innovation, and interconnectedness, a reminder that even the smallest organisms can have monumental impacts on the planet's trajectory. Every organism that swims in today's seas, every coral reef, every planktonic bloom, carries the legacy of these earliest aquatic pioneers. From these humble beginnings, life began its inexorable journey, slowly mastering the water, then the land, and eventually the skies. The oceans, in their vastness, not only gave life its first home, but also its first challenges, first victories, and first opportunities to evolve into the rich tapestry of existence we witness today. The first microbe were the pioneers of biology, the earliest forms of life capable of growth, reproduction, and adaptation. Unlike the protocells that preceded them, these organisms had developed stable metabolic pathways, allowing them to extract energy from their surroundings efficiently. Some thrived on chemical compounds seeping from volcanic vents, while others absorbed sunlight and carbon dioxide to fuel photosynthesis. In every sense, they were masters of survival, perfectly suited to the extreme conditions of early Earth. These microbes were astonishingly simple, yet extraordinarily versatile. Encased in protective membranes, they contained genetic material capable of replication and mutation. This genetic blueprint allowed them to adapt to changing conditions, experiment with new metabolic strategies, and colonize virtually every niche in the oceans. Their ability to reproduce and evolve laid the foundation for all future life, from the tiniest plankton to the largest mammals. Fossil evidence gives us glimpses of these ancient pioneers. Stromatolites, layered rock formations created by microbial mats, are among the earliest signs of life on Earth. Dating back more than 3.5 billion years, these structures demonstrate not only the existence of microbes, but also their incredible capacity for cooperation. In colonies, they trapped sediment, produced oxygen, and modified their environment, creating conditions that would enable more complex life to flourish. The first microbes were also the architects of one of Earth's most transformative events, the Great Oxidation Event. By performing photosynthesis, cyanobacteria began producing oxygen as a byproduct, slowly enriching the atmosphere and oceans. This increase in oxygen concentration paved the way for the evolution of aerobic organisms, dramatically expanding the possibilities for life on the planet. Without these early microbes, the rich diversity of life that we observe today, including humans, would never have been possible. Life at this stage was a delicate balance between opportunity and danger. Microbes had to navigate fluctuating temperatures, ultraviolet radiation, chemical gradients, and predatory threats from other microorganisms. Those that adapted survived. Those that did not vanished without a trace. It was a relentless, natural, selection-driven process that tested the resilience and ingenuity of life at its inception. Consider the scale of this achievement. From a world dominated by chemical reactions and barren waters, these microscopic entities emerged, capable of self-replication, energy production, and environmental modification. Each microbe was a miniature laboratory, performing thousands of reactions per second, testing molecular combinations and refining survival strategies. Through countless generations, these processes led to the emergence of increasingly sophisticated life forms, culminating in multicellular organisms. Microbes were not merely passive inhabitants of early Earth. They were active agents of change. By modifying their environment, producing oxygen, forming mats, and influencing chemical cycles, they transformed the planet itself. Their existence demonstrates a profound truth. Life, even in its simplest form, has the power to shape worlds. The first microbes were also the ancestors of every organism alive today. From the smallest bacteria to the most complex mammals, all life carries within it echoes of these early pioneers. Every adaptation, every metabolic pathway, every cellular process can trace its origins back to these tiny architects of life. They were the first to harness energy, to replicate with fidelity, and to adapt to an ever-changing world. In contemplating these first microbes, we are reminded of the resilience, creativity, and tenacity inherent in life. What seems insignificant at first glance, the smallest, simplest organism, can, over billions of years, transform an entire planet. The story of the first microbe is not just a story of survival. It is the story of life's ingenuity, persistence, and its relentless drive to flourish against all odds. The history of Earth is a history of transformations, and one of the most pivotal shifts occurred when life moved from the water to the land. While microscopic organisms had already colonized the oceans, the emergence of plants on land marked a dramatic turning point in the story of life. The first plants were not the towering trees and sprawling forests we see today, but small, simple organisms that began to establish themselves on damp soils, rocks, and the edges of shallow water. They were pioneers in an uncharted terrestrial world, facing the challenges of sunlight, desiccation, and fluctuating temperatures. These early plants were likely bryophyte-like organisms, similar to modern mosses and liverworts, with simple structures capable of absorbing water directly from the environment. They lacked roots, stems, or complex vascular systems, but were highly effective at surviving in moist habitats. Their simple yet adaptable forms allowed them to exploit niches that no other organism had occupied, gradually spreading across the newly forming landmasses. One of the most critical innovations of these first plants was photosynthesis. By converting sunlight into chemical energy, they could sustain themselves while producing oxygen as a byproduct. This process had profound implications not only for the plants themselves, but also for the entire planet. The oxygen released into the atmosphere began to accumulate, slowly transforming Earth's environment and setting the stage for the evolution of aerobic life. Without these early photosynthetic pioneers, the rich diversity of life that would follow could not have existed. The structure of the first plants reflected their adaptation to the challenges of terrestrial life. Small, flat structures maximized surface area for sunlight absorption, while simple anchoring systems allowed them to remain attached to rocks and soil. Though fragile by modern standards, these organisms were resilient and highly effective at colonizing new habitats. Through natural selection, variations that allowed better water retention, sunlight capture, or reproductive efficiency were favored, leading to gradual evolutionary advancements. As these plants proliferated, they began to shape the environment itself. By stabilizing soil and capturing carbon dioxide, they contributed to the development of a more hospitable terrestrial ecosystem. The oxygen produced through photosynthesis not only enriched the atmosphere, but also facilitated the eventual rise of complex organisms, including animals. Over millions of years, these simple plants paved the way for more complex vascular plants, the formation of the first forests, and the establishment of ecological niches that would support a diversity of terrestrial life. Fossil evidence, including early spores and plant impressions in ancient rock formations, provides insight into the proliferation of these early plants. These fossils demonstrate that even the smallest organisms can exert a profound influence on their environment. By modifying the chemistry of soils, interacting with microbial communities, and altering atmospheric composition, the first plants set the stage for the complex web of life that would develop on land. The colonization of land by plants also had a cascading effect on other life forms. By providing food and habitat, these pioneers created opportunities for invertebrates and later, more complex animals to venture onto land. The establishment of terrestrial ecosystems was gradual, yet the first plants were indispensable architects of this transformation. Their presence created a framework for life to diversify, adapt, and thrive in ways that would have been impossible in their absence. In contemplating the first plants, one can appreciate the interplay between life and environment. These organisms, small and seemingly insignificant at first, had the capacity to alter the course of Earth's history. They were the original environmental engineers, modifying the landscape, stabilizing ecosystems, and generating oxygen that would support the evolution of all aerobic life. Every tree, shrub, and grass blade we see today owes its ancestry to these humble pioneers. The story of the first plant is the story of persistence, adaptation, and innovation. It illustrates how life, even in its simplest form, can exert enormous influence, shaping the planet and creating conditions for future complexity. The emergence of terrestrial plants was a turning point in the history of life, a moment when Earth became not just a world of water and microbes, but a planet capable of supporting rich, diverse ecosystems on land. Photosynthesis. The story of life on Earth is inseparable from the story of photosynthesis, a process that transformed the planet and enabled life to flourish on an unprecedented scale. Before photosynthesis, the atmosphere contained only trace amounts of oxygen, and energy flow in ecosystems was limited to chemical reactions occurring in water or in close association with geothermal vents. The emergence of photosynthetic organisms marked a revolutionary turning point. Life had discovered a way to harness the energy of the sun, converting it into chemical power and altering the chemical and physical landscape of the planet forever. The earliest photosynthetic organisms were microscopic cyanobacteria, sometimes called blue-green algae. These microorganisms were astonishingly efficient at capturing sunlight and converting carbon dioxide and water into sugars, releasing oxygen as a byproduct. For the first time in Earth's history, life began producing a gas that would accumulate in the atmosphere, creating a foundation for the evolution of aerobic organisms. Cyanobacteria did not merely survive, they reshaped the world around them. Their mats formed extensive stromatolites, layered structures that entombed sediments and became some of the earliest fossil evidence of life. Photosynthesis was more than just a chemical process. It was an innovation that enabled life to scale up. The energy captured from sunlight allowed these microorganisms to reproduce more rapidly, support larger colonies, and diversify metabolic pathways. In ecosystems previously limited by the availability of chemical energy, the introduction of photosynthesis created new niches and opportunities. Oxygen, once toxic to many anaerobic organisms, became a catalyst for evolutionary innovation, allowing the development of more complex life forms that relied on oxygen for respiration. The process itself is a marvel of biological engineering. Within specialized cellular structures called thylakoids, light energy is absorbed by pigments like chlorophyll. This energy drives the conversion of carbon dioxide and water into glucose, a molecule storing both energy and chemical potential. Simultaneously, oxygen is released into the surroundings, gradually increasing the concentration of this life-sustaining gas in the oceans and atmosphere. The balance of energy capture, storage, and environmental transformation created a feedback loop that accelerated the proliferation of photosynthetic life. Over hundreds of millions of years, photosynthetic organisms diversified and spread across the planet. Initially confined to shallow waters where sunlight could penetrate, these organisms gradually colonized new environments, developing adaptations that allowed them to survive in fluctuating light conditions, varying temperatures, and changes in water chemistry. The early oceans, once dominated by chemical gradients, became hubs of biological productivity, teeming with microbial life supported by the energy captured through photosynthesis. The oxygenation of Earth's atmosphere, driven by photosynthesis, had profound geological and biological consequences. Oxygen combined with iron in the oceans to form banded iron formations, altering sedimentary rock structures. On land, the rise in atmospheric oxygen contributed to the formation of the ozone layer, which shielded the surface from harmful ultraviolet radiation and allowed life to venture safely onto dry land. Photosynthesis, therefore, was not just a metabolic process. It was a planetary force, fundamentally altering both chemistry and climate. The significance of photosynthesis extends beyond energy production. It set the stage for ecological interactions, food webs, and the complexity of multicellular life. Plants, algae, and cyanobacteria became the primary producers, generating the organic matter that would feed herbivores, predators, and decomposers for billions of years. Every organism that relies on oxygen and organic compounds for survival owes its existence to the innovations of photosynthetic pioneers. Consider the elegance of this system. Life discovered a way to transform sunlight, a seemingly intangible resource, into matter, energy, and environmental change. This innovation enabled life to escape the constraints of purely chemical energy sources, expand into new habitats, and build complexity upon complexity. The first oxygen-rich ecosystems were laboratories of evolution, where competition, cooperation, and adaptation drove the emergence of multicellular organisms, complex marine ecosystems, and eventually terrestrial plants and animals. Photosynthesis also exemplifies the interdependence of life and environment. As photosynthetic organisms altered the chemistry of water and air, they created conditions for new forms of life to evolve. Each wave of oxygenation, each colonization of new ecological niches, triggered feedbacks that shaped the trajectory of life on Earth. This interplay between biology and planetary chemistry continues to this day, highlighting the fundamental role of photosynthesis in maintaining the balance of ecosystems and climate. The story of photosynthesis is a story of innovation, resilience, and planetary transformation. From the first cyanobacteria floating in shallow waters to the vast forests and algal blooms that cover the Earth today, this process has been a constant driver of life's diversity and abundance. It reminds us that even microscopic organisms, through ingenuity and persistence, can wield influence on a planetary scale, shaping the conditions for life across billions of years. The first animal, the emergence of the first animal, represents one of the most dramatic leaps in the history of life on Earth. While unicellular organisms and early plants had already transformed the oceans and atmosphere, the appearance of multicellular animals introduced movement, predation, and complex interactions that would redefine ecosystems. These early animals were small, simple, and often microscopic, yet they were the pioneers of an entirely new mode of life, one capable of sensing, responding, and actively navigating its environment. The first animals likely evolved from colonies of specialized cells. These clusters of cells, originally living in cooperation for survival, gradually developed division of labor. Some cells specialized in reproduction, others in feeding, and still others in basic movement. This cellular differentiation marked the birth of multicellularity, a process that would eventually allow for the incredible complexity observed in modern animal life. Among the earliest known animals are the sponges, simple organisms that remain largely sessile but possess coordinated systems for water flow, feeding, and waste removal. Sponges demonstrate the remarkable potential of multicellular organization, where individual cells communicate and function as a collective organism. Despite their simplicity, these first animals were capable of filtering nutrients from water, interacting with their environment, and reproducing efficiently, laying the foundation for the evolutionary explosion that would follow. The evolution of the first animal also had profound ecological consequences. Their feeding activities reshaped microbial communities, influencing the distribution and abundance of other early life forms. Predation, even at this microscopic level, introduced selective pressures that accelerated evolution, encouraging adaptations in both predator and prey. This dynamic interplay drove diversification, promoting the emergence of novel forms and complex life cycles. Fossil evidence provides crucial insights into these early animals. The Ediacaran biota, dating back approximately 600 million years, includes impressions of soft-bodied organisms that exhibit the earliest known multicellular structures. These fossils reveal the morphology and organization of primitive animals and hint at the early experimentation in body plans, symmetry, and movement that would characterize future evolutionary innovations. Movement was one of the most significant innovations introduced by these first animals. Even minimal mobility allowed organisms to explore new habitats, escape unfavorable conditions, and actively acquire resources. This capacity to move set the stage for predation, competition, and complex ecological networks that would dominate the oceans for hundreds of millions of years. Another crucial development was the emergence of sensory structures, even in their simplest form. Early animals evolved the ability to detect chemical signals, light, and physical stimuli. This sensory capability provided an evolutionary advantage, guiding feeding behavior, reproduction, and environmental interactions. The integration of sensory input with movement marked a pivotal step toward behavioral complexity. foreshadowing the intricate nervous and muscular systems of later animals. Reproduction in these early animals involved both sexual and asexual methods, allowing populations to expand rapidly and maintain genetic diversity. This flexibility enhanced adaptability, ensuring survival in fluctuating environmental conditions. Over time, sexual reproduction became increasingly dominant, introducing greater genetic variation and accelerating evolutionary innovation. The appearance of the first animal also influenced the development of early ecosystems. By consuming microbes, filtering water, and interacting with their surroundings, these organisms initiated the first trophic interactions. They were no longer passive participants in the environment. They became active engineers, modifying habitats, influencing nutrient cycles, and creating conditions for future species to evolve. The significance of the first animal extends beyond biology. It represents a conceptual liptha ability of life to move, interact, and shape its environment. Multicellularity, sensory perception, and mobility set the stage for the incredible diversity of life forms that would follow, from simple invertebrates to complex vertebrates, and eventually humans. The first animal embodies the transition from simple cellular assemblies to organized, responsive, and adaptive life capable of influencing ecosystems on a grand scale. In contemplating the first animal, one sees the profound consequences of innovation and collaboration at the cellular level. Life, which began as solitary, microscopic entities, had now discovered the power of unity, specialization, and active interaction. These early animals were small, simple, and fragile, yet they carried the blueprint for all future animal life, representing a transformative moment in the story of Earth's biosphere. Marine life. The oceans of early Earth were once silent, dark expanses, populated only by microscopic organisms. Over millions of years, these waters transformed into thriving ecosystems as multicellular animals emerged and diversified. Marine life became the driving force of ecological complexity, innovation, and evolutionary experimentation. The oceans were not just a habitat, they were laboratories where life tested new forms, interactions, and survival strategies, shaping the trajectory of the planet's biodiversity. Early marine ecosystems were dominated by soft-bodied invertebrates, sponges, and primitive worms. These organisms established the first food webs, with some filtering microscopic particles from the water, while others grazed on microbial mats or scavenged decaying matter. This early trophic structuring introduced dynamics of energy flow and nutrient cycling, setting the foundation for the intricate marine ecosystems that would evolve over hundreds of millions of years. Fossil evidence from the Ediacaran and Cambrian periods reveals the diversity and complexity of early marine life. The Cambrian explosion, approximately 541 million years ago, marked a remarkable evolutionary surge in which virtually all major animal phyla appeared. Creatures with hard shells, exoskeletons, segmented bodies, and specialized appendages began to populate the seas. This period illustrates how environmental stability, increased oxygen levels, and ecological interactions can accelerate evolutionary innovation. Predation became a powerful evolutionary force in these early oceans. The development of shells, spines, and defensive mechanisms among prey organisms drove adaptive responses, while predators evolved strategies for hunting, sensing, and mobility. This evolutionary arms race fostered rapid diversification, pushing marine life toward increasingly sophisticated forms and behaviors. The oceans became arenas of competition, cooperation, and survival, where life constantly innovated to thrive. Photosynthetic organisms continued to play a crucial role in marine ecosystems. Algae and cyanobacteria formed the primary producers, generating the oxygen and organic matter necessary to sustain higher trophic levels. Coral-like organisms began constructing reefs, creating complex habitats that supported diverse communities of invertebrates, fish, and other marine species. These early ecosystems were highly dynamic, influenced by currents, nutrient availability, and geological processes such as volcanic activity and continental shifts. The structure of early marine life also evolved in response to environmental pressures. The development of segmentation, appendages, and sensory organs allowed animals to navigate, capture food, and respond to threats more effectively. Early eyes, tentacles, and antennae introduced a level of environmental awareness that had never existed before, enabling animals to interact with their surroundings in increasingly sophisticated ways. Movement and behavior became critical factors in survival, driving the emergence of complex life strategies. The oceans also facilitated dispersal and colonization. Currents, tides, and waves allowed larvae and juvenile forms to spread over vast distances, promoting genetic diversity and reducing competition in localized areas. This mobility enabled species to explore new habitats, adapt to varying conditions, and ultimately evolve into distinct lineages that would populate every corner of the seas. Marine life was not static. It continually reshaped the ocean environment. Organisms contributed to the cycling of nutrients, the formation of sediments, and even the chemistry of the oceans. Coral reefs, microbial mats, and burrowing animals modified habitats, influencing the distribution and abundance of other species. Life and environment co-evolved in a continuous feedback loop, with each new adaptation creating opportunities and challenges for other organisms. The complexity of marine ecosystems also set the stage for evolutionary innovation on land. Many features essential for terrestrial life, such as respiration, locomotion, and sensory perception, first developed in aquatic environments. Fish-like vertebrates, for example, evolved fins, gills, and primitive lungs that would later facilitate the colonization of land. In this way, the oceans were both a crucible and a bridge, nurturing life while preparing it for the next great frontier, the terrestrial world. Studying early marine life provides a profound insight into the principles of evolution, adaptation, and ecological interdependence. From microscopic plankton to the first armored invertebrates, the oceans demonstrate how life can diversify, specialize, and construct complex ecosystems. Each organism, whether predator or prey, stationary or mobile, contributed to the ongoing story of life, demonstrating the interconnectedness and resilience of biological systems. The story of marine life is a testament to the transformative power of oceans. These vast, nutrient-rich environments not only sustained early life, but also fostered innovation, diversity, and complexity on a planetary scale. The creatures of the ancient seas laid the foundation for future ecosystems, shaping the evolutionary pathways that would eventually lead to land animals, birds, and mammals, including humans, pre-human creatures. The story of life on Earth takes a dramatic turn with the emergence of pre-human creatures, organisms that would, eventually, pave the way for the appearance of Homo sapiens. These creatures evolved over millions of years, inhabiting diverse environments, experimenting with new forms, and developing adaptations that would later define mammalian and primate evolution. Pre-human life was a bridge between the ancient oceans and plants and the complex terrestrial ecosystems capable of supporting advanced intelligence. In the vast expanse of prehistoric Earth, reptiles, amphibians, and early mammals dominated terrestrial landscapes. Each group represented evolutionary experimentation, exploring different strategies for survival. Amphibians, descendants of fish, had already adapted to life on land, developing lungs, limbs, and sensory organs capable of navigating both water and solid ground. Their bodies reflected a dual existence, bridging the aquatic origins of vertebrates with the potential for terrestrial colonization. Early reptiles demonstrated further innovation, with stronger skeletal structures, scaly skin to reduce water loss, and eggs that could survive on land. Reptiles became increasingly independent of aquatic environments. They diversified rapidly, occupying niches that included predators, herbivores, and scavengers. These adaptations not only allowed reptiles to thrive, but also created selective pressures that would influence the evolution of mammals and, eventually, primates. The first mammals appeared in the shadows of dominant reptiles, small and nocturnal, relying on keen senses of smell and hearing to navigate a world filled with predators. These early mammals were the ultimate survivors, adaptable and resilient. Their ability to regulate body temperature, care for offspring, and exploit new ecological niches allowed them to persist through changing climates, mass extinctions, and the rise and fall of dominant species. Mammals were modest in size, yet their evolutionary potential was vast, providing the blueprint for future complexity. Among mammals, the primate lineage began to emerge, displaying a remarkable combination of dexterity, sensory perception, and social behavior. Early primates evolved grasping hands, forward-facing eyes for depth perception, and larger brains relative to body size. These adaptations facilitated climbing, hunting, and complex interaction within social groups. In forests and woodlands, primates became highly efficient foragers and explorers, laying the groundwork for cognitive and cultural evolution that would later define humans. Pre-human creatures also included a wide range of other vertebrates, such as early birds, marsupials, and large herbivores. Birds evolved from small, feathered theropod dinosaurs, mastering flight and revolutionizing ecological niches. Large herbivores shaped landscapes through grazing and seed dispersal, influencing plant evolution and ecosystem structure. These interactions highlight the intricate web of life, where each organism contributes to environmental modification, evolutionary pressure, and ecosystem stability. Fossil evidence offers a window into this fascinating era. Skeletons, teeth, and footprints reveal the physical adaptations of pre-human creatures, while traces of behaviors such as burrows, nests, and social groupings illuminate how these animals interacted with their environment and each other. Paleoecological studies further demonstrate how climate shifts, geological events, and predator-prey dynamics influenced evolutionary trajectories, driving the emergence of traits that would eventually characterize the human lineage. The transition from pre-human creatures to hominins involved numerous evolutionary experiments. Bipedalism, tool use, and advanced cognition did not appear suddenly, but were the result of incremental changes over millions of years. Pre-human creatures exhibited varying degrees of these traits, providing a stepping stone for the development of complex intelligence and culture. Their survival and adaptation in dynamic environments exemplify the principles of natural selection and the power of evolutionary innovation. By studying pre-human creatures, scientists gain insight into the forces that shaped human evolution. Morphological adaptations, behavioral strategies, and ecological interactions all provide clues about the challenges and opportunities that influenced the trajectory from early mammals to intelligent primates. These organisms demonstrate that the emergence of humans was neither random nor isolated. It was the culmination of a long, intricate process of biological experimentation, adaptation, and survival. Pre-human creatures also remind us of the interdependence of life and environment. Evolutionary success depended on the ability to respond to climate shifts, resource availability, and competition with other species. The development of social structures, communication, and cooperative behaviors among some pre-human species foreshadowed the complex societies that humans would eventually build. In every sense, these creatures laid the foundations of human potential. The story of pre-human creatures is a story of resilience, innovation, and the gradual building of complexity. Each adaptation, each evolutionary experiment contributed to a lineage that would eventually produce humans. These creatures bridged the ancient past with the future, demonstrating how incremental change, environmental interaction, and survival drive the evolution of intelligent life. Their legacy is embedded in every human trait, from anatomy to cognition, from social behavior to culture, providing a living connection to Earth's deep biological history. The formation of DNA at the core of every living organism lies an extraordinary molecule, DNA, deoxyribonucleic acid. The formation of DNA represents one of the most profound innovations in the history of life on Earth. Long before the emergence of multicellular organisms, plants, or animals, the basic blueprint for life itself was being written at a molecular level. DNA's complex structure and unparalleled capacity for storing information made it the foundation upon which all biological complexity would be built. The origins of DNA are closely intertwined with the chemical environment of early Earth. In primordial oceans, rich with organic molecules and energized by lightning, ultraviolet radiation, and hydrothermal vents, basic components like nucleotides began to form. Nucleotides, consisting of a sugar, phosphate, and nitrogenous base, represented the first building blocks of genetic material. Over time, these molecules linked together, forming short chains that carried rudimentary information, a prelude to the intricate coding systems that would emerge billions of years later. RNA, a chemically similar molecule, likely preceded DNA as the first carrier of genetic information. RNA's ability to both store information and catalyze reactions made it a versatile molecule capable of sustaining early life through a series of evolutionary refinements. RNA molecules began to replicate more accurately, paving the way for the eventual transition to DNA. Unlike RNA, DNA offered a more stable, durable, and efficient medium for long-term storage of genetic information, allowing life to sustain increasingly complex structures and functions. The formation of the double helix, a defining feature of DNA, was a turning point in evolutionary history. The helical structure, with complementary base pairing between adenine and thymine, guanine and cytosine, provided a mechanism for accurate replication and repair. This molecular precision ensured that genetic information could be faithfully transmitted from one generation to the next, preserving traits and enabling the slow accumulation of evolutionary innovations. DNA became the repository of heredity, the molecular ledger of life's history, and the blueprint for all cellular machinery. The emergence of DNA also allowed for the development of genetic codes, regulatory mechanisms, and complex biochemical pathways. Proteins, which perform virtually every function in living organisms, are synthesized according to instructions encoded in DNA. The interplay between DNA, RNA, and proteins created the central dogma of molecular biology, a framework that governs the growth, reproduction, and evolution of all living cells. This molecular system is both remarkably resilient and incredibly versatile, capable of supporting life forms as simple as bacteria and as complex as humans. As organisms evolved, DNA facilitated adaptation and innovation. Mutations, changes in the sequence of nucleotides introduced variation within populations. Natural selection acted upon this variation, favoring traits that enhanced survival and reproduction. Over billions of years, the accumulation of such molecular changes led to the astonishing diversity of life on Earth. From microscopic microbes to sprawling forests, coral reefs, and intelligent animals, DNA is thus not merely a molecule. It is the engine of evolution, recording life's history while simultaneously driving its future. The structural elegance of DNA also allows it to interact with environmental and cellular factors. Epigenetic mechanisms, for instance, modify gene expression without altering the underlying sequence, enabling organisms to respond dynamically to environmental challenges. These regulatory systems illustrate the sophistication of DNA as more than a static code. It is a responsive, adaptive blueprint capable of integrating signals from the environment, coordinating cellular processes, and guiding development. Fossilized molecules, molecular clocks, and comparative genomics provide insight into the ancient origins of DNA and its role in early life. By comparing genetic sequences across species, scientists can trace lineages back billions of years, reconstructing evolutionary pathways and illuminating the molecular innovations that enabled life to thrive. This research demonstrates that DNA was central not only to survival, but also to the diversification and complexity of all living organisms. The formation of DNA set the stage for every subsequent milestone in life's history. It enabled the rise of multicellular organisms, the colonization of land by plants and animals, and eventually the evolution of intelligence and culture. DNA's capacity for precise replication, variation, and regulation allowed life to experiment, innovate, and adapt to changing environments across geological epochs. Every organism, from the smallest bacterium to humans, carries within it the legacy of this molecular revolution. Understanding the formation and function of DNA underscores the profound interconnectedness of all life. It reveals how a molecule, born in the primordial oceans, could orchestrate the complexity of ecosystems, drive evolution, and ultimately give rise to consciousness capable of contemplating its own origins. DNA is the ultimate testament to the power of chemistry and evolution, a bridge between the abiotic world of molecules and the rich, diverse biosphere we inhabit today.